Welcome to Endocrine and Reproductive System. This is the thyroid hormone synthesis, function, and control, and I'm Lynn. Let's go ahead and get started with this module. In this session, we're going to cover the following learning objectives. Identify the steps in biosynthesis, storage, and secretion of T3 and T4 and their regulation. Describe the molecular basis of thyroid hormone synthesis. Discuss the regulation of iodine uptake by the thyroid gland and the effects of iodine deficiency. Describe the absorption, uptake, and distribution and excretion of iodide. Explain the importance of thyroid hormone binding in blood on free and total thyroid hormone levels. Explain the significance of conversion of T4 to T3 and reverse T3, also known as RT3, in extrathyroidal tissues. Explain the regulation of thyroid hormone synthesis by thyrotropin-releasing hormone TRH and thyroid-stimulating hormone TSH. Describe the physiological effects and mechanisms of action of thyroid hormones. We'll delineate the causes and consequence of over and under secretion of thyroid hormone. Explain the metabolic functions, effects, and regulation of thyroid hormones. We'll discuss the conditions that can cause enlargement of the thyroid gland. Discuss the etiology of common diseases that cause hyper and hypothyroidism and their common presentations and treatments. Synthesis and secretion of thyroid metabolic hormones. The thyroid gland is the largest body structure devoted entirely to endocrine activity. The thyroid gland is a butterfly-shaped gland located immediately inferior to the thyroid cartilage, right at the anterior aspect of the trachea. It is covered by this connective tissue that is capsule that has both a left and right lobe, and it generates this reddish tint color. The thyroid gland is composed of many microscopic spherical structures called the thyroid follicles. T4 and T3, thyroxine and triiodothyronine, are the active hormones produced by this gland. On average, there's about 35 micrograms of T3 produced per day. Almost all of T4 is converted to T3. T3 is 4 times as potent as T4. It has a much shorter duration, and it can be stored in the follicles inside the thyroid gland to 2 to 3 months. These follicles store thyroglobulin, Tg, which is a substrate for creating T3 and T4. If you look closely at the histology, it is composed of cuboidal epithelial tissue and cells. On the right side, you can see the structure. The wall of each follicle is formed by the simple cuboidal lining of epithelial tissue. The central lumen of these bubbles are filled with what we call colloid. The follicular cells are able to produce the thyroid hormone. This is done by synthesizing the intermediate TGB, the thyroglobulin protein, which is secreted inside the colloid fluid. On a posterior aspect, you can see that there are 4 small glands located on the back side of the thyroid gland, and these are called the parathyroid glands. The region that divides the left and right lobes is called the isthmus, and you can see that it sits right below the thyroid cartilage. The formation and secretion of thyroglobulin by the thyroid cells. The thyroid cells are typical protein-secreting glandular cells. The endoplasmic reticulum and the Golgi apparatus is able to secrete into the follicles large amount of this protein called thyroglobulin. It has a molecular weight of about 300,000 daltons, and each of these thyroglobulin contains numerous tyrosine amino acid residues. These are the important substrates that are used to combine with iodine to form the thyroid hormone. And thus, that's why the thyroid hormone is formed from the thyroglobulin molecule. So think of it like this. T4 and T3 hormones are formed from tyrosine combined with parts of the thyroglobulin molecule during this process inside the follicular colloid. The oxidation of iodide ion is the first step. It starts by consuming iodide ions and it gets oxidized into a form called I3 negative. Binding of iodine with thyroglobulin protein is a process known as organification. It requires an enzyme called a thyroid peroxidase that causes this to process. It occurs about within seconds to minutes, but tyrosine is critical and iodine is critical to create into the first form called monoiodotyrosine and then converted to a 2-iodine compound called diiodotyrosine. Tyrosine going through the peroxidase enzyme, becoming monoiodotyrosine. Over here you can see the I being attached to it. Then it gets converted into the diiodotyrosine, and then when you have one mono and one di, you can create a tri, right? 1 plus 2 is 3, so you have a T1, T2 becoming T3. And then there's another intermediate called the triiodothyronine, RT3, which does not play any significance in humans. The peroxidase system could be genetically absent or potentially blocked, which can cause a patient to not be able to produce any of these hormones. Very small amounts of the reverse T3, RT3, is formed in this process of coupling T1 and T2. RT3 does not have any significance in the human system, so it's not readily tested. The final form over here you can see is T4, and again you can see there are 4 iodines on the rings. Sodium iodide symporter, or also known as iodide trapping. Most of the thyroglobulin is not released into the circulating blood. Instead, our T4 and T3 are cleaved from that thyroglobulin molecule, and then these free-form hormones are released. This is the process and step-by-step formation of the thyroid hormone. Step number 1, we transport the ingested iodides from the blood into the ingested thyroid follicles. Step number 2, the basal membrane pump is going to then utilize a sodium iodide symporter where it can transport 1 iodide with 2 sodium ions into the cell. The first hormone that begins releasing from the pituitary gland, called the TSH, thyroid stimulating hormone, is going to trigger and increase the synthesis and concentration of iodide in the cell. This process of stimulating the uptake of iodide is called iodide trapping. As iodide is oxidized into the iodine form via the thyroid peroxidase, it diffuses to the follicular lumen. Iodine atoms are covalently attached then to the tyrosine residues on that thyroglobulin protein. This will then create the MIT T1 or DIT T2. 2 DIT molecules can form T4, and then 1 MIT and then 1 DIT can form T3. Then the follicular cells will uptake these iodinated thyroglobulin via endocytosis. There is some iodine that gets recycled in this process, which we'll talk about later in this module. T4 is then released into the blood, then is bound to proteins, generally bound to the thyroid binding globulin or albumin. On the right side, you can see this is an image showing you thyroglobulin is Tg, iodine is I, MIT is monoiodotyrosine, and DIT is diiodotyrosine, and then we have the iodinated thyroglobulin, and on the bottom we have T4 and T3. These are 2 kinds of transports. In order to power this pump, we require an ATPase, a sodium potassium pump, to build up a concentration gradient. This symporter pumps the sodium and the iodide into the follicle cell. The thyroid peroxidase is going to then oxidize the iodide to iodine. Over here on the left, the TSH receptor binds to the TSH hormone. This will then trigger the binding combination of thyroglobulin with the oxidized iodine. That will then create either the MIT or the DIT, and then you can combine it in creating the T3, T4. Then it goes back into the follicular cell via the enzymes for modification, and then the T3 and T4 are created, which are then bound to proteins and transported in your bloodstream. So you can see that it begins from transportation of iodide and iodine from the blood to the follicular cells, up to the follicular lumen, from the follicular lumen back down into the blood vessels. Release of thyroxine and triiodothyronine from the thyroid gland. The thyroxine and triiodothyronine are cleaved immediately. They're processed into release of T3, T4. The thyroglobulin in the colloid enters the cells by binding to another transport protein called megalin, and the diffusion occurs through the base of the thyroid cell into the surrounding capillaries. Keep in mind that 99% of all the T3, T4 is bound. Most of it is bound to the thyroxine binding globulin, or the TBG. There's a lesser extent that's bound to albumin, but you can still find traces of T3, T4 in albumin. Here's a comparison of half-life. T3, T4 are slowly released to tissue because of the affinity that it has to these plasma binding proteins. T4 has a half-life of about 6 days, and T3 has a half-life of about 1 day. Here's a clinical application. The iodinated tyrosines are then cleaved by the deiodinase enzyme that makes iodine available again for recycling. So not all the iodine is wasted. Your body does recycle some of this. And if there is a deficiency or mutation or some sort of a damage to the deiodinase enzyme, you might also have an issue in creating T3, T4. Transport of T3 and T4 to tissues. The thyroid hormones have a slow onset but a long duration of action. A long latent period before the thyroxine activity begins, so it reaches a maximum peak at about 10 to 12 days and decreases with a half-life of about 15 days. Approximately, you can see that when you do an experiment, within the first 9 to 10 days, the vasometabolic rate boosts up about 5 to 10 folds. It maxes out right at day 10, and then after day 10, it begins to slope downwards, decreasing over the course of time. There is a slight latency and prolonged period of action because these hormones are so tightly bound to their globulin proteins inside the blood plasma, and it takes some time for them to unload off. The T3 occurs about 4 times as rapid and more potent than T4. Physiological functions of the thyroid hormone. Thyroid hormones increase transcription of large number of genes. Most of the T4 secreted by the thyroid is converted into T3. Before acting on genes to increase genetic transcription, one iodide is removed from almost all of the T4. This will then create the T3, triiodothyronine. The intracellular thyroid hormone have receptors that have a high affinity for T3. And this is why more than 90% of all the thyroid hormone binds with this receptor is going to be T3 based. The thyroid hormone receptors are located attached to genetic strands or located in proximity to the DNA. It is what we call a heterodimer, which means 2 parts that are different. They bind with the retinoid X receptor, also known as the RXR. This is a very specific thyroid binding response element on DNA, which will trigger a numerous numbers of outcomes. It can increase protein synthesis in growth of many kinds of cells. It can increase central nervous system development. And you can look at the list for the cardiovascular increasing activity. It increases cardiac output, increases blood flow, increases heart rate, increases respiration rate, increases heart strength. It also boosts a positive increase in all of metabolic pathways, increasing mitochondrial activity, speeding up the NAK ATPase, increasing the oxygen consumption, increasing glucose absorption, increasing gluconeogenesis, increasing glycogenolysis, increasing lipid metabolism, and increasing protein synthesis, and of course BMR. There are also some non-genomic thyroid hormone actions. These are typically located on the plasma membrane and cytoplasma, and sometimes affecting the mitochondria. It regulates some of the ion channels of certain cells, and of course increasing the cyclic AMP, the secondary messenger, located inside our cells. T3, T4 coming in. T4 requires the iodinase enzyme to be converted to T3. T3 is the active binding, 90% of it binds to the receptor. And there's 2 parts. There's a thyroid binding domain, and there is the retinoid X receptor domain as well. Both of these will then latch onto a specific segment of the DNA and boost genetic transcription. Thyroid hormones increase cellular metabolic activity. Metabolic activity increases up to about 60 to 100% above normal. And this is why there's going to be substantial weight loss and catabolism in folks that have hyperthyroidism. Thyroid hormones increase the number and activity of the mitochondria. As we know, the mitochondria is the powerhouse of the cell, and most cells will increase in size and number. The total membrane surface area is also going to be increased. This means that you're going to generate a lot more ATP in a shorter span of time. The thyroid hormone also increases the active transport of many ions. This is particularly the case for NAK pump. It causes cells to become even more leaky to sodium and potassium. And it also will promote growth and development for the fetal brain in early stages of the life. So let's talk about some of the very specific effects the thyroid hormone has on different parts of the body. In the next several slides, we're going to go over specific areas of the body and what exactly does the thyroid hormone do and how does it act. Stimulation of carb metabolism. The thyroid hormone has a positive impact on the ability to metabolize carbohydrates. It increases the glucose uptake inside our cells. It enhances glycolysis and enhances the gluconeogenesis. It also increases the rate by which the amount of nutrients are absorbed in the GI system, and it increases the release of insulin. It also has a stimulation impact on fat metabolism, accelerates oxidation of fatty acids by cells. There's also an effect on the plasma and liver fat. An increase in thyroid hormone will decrease the concentration of cholesterol, phospholipids, and triglycerides in the plasma because there's a faster and higher metabolic rate, depleting them from the blood system. It also plays a role to remove deposition of fat from the liver, so there's more secretion via the bile juices and excretion of waste. This is also why in prolonged hypothyroidism, there is going to be a slower metabolism of cholesterol and lipids, resulting in potential accumulation in the blood vessels, leading to atherosclerosis and the plaque buildup. There's also an increase in the cholesterol secretion in bile, and hence there's a consequent loss of it in the feces. It also increases the requirements for body vitamins because metabolic goes up. The thyroid hormone could increase the demand for vitamin, leading into potential vitamin deficiencies because there's a higher need to maintain this kind of BMR. It increases the BMR up to about 60 to 100% above normal. It will decrease your body's weight because you are utilizing a lot more of the macronutrients, and almost results in a decrease of body weight. There's a positive impact on the increasing of blood flow and cardiac output, and thus vasodilation to most body tissues, increasing blood flow. Blood flow to the skin is also increased because there is a need to eliminate the amount of heat generated. It also increases the strength of the heart's contraction and heart rate. Excessive amounts of thyroid hormone could even lead to a cardiac depression because your heart is overloaded. There's also going to be a change to the arterial blood pressure. The mean arterial pressure usually maintains about normal, but the pulse pressure and the systolic pressure can be elevated by up to 10 points in hyperthyroidism. An increase in thyroid hormone will lead to an increasing of respiration. The increase of metabolism is going to require a higher amount of oxygen usage, and therefore a higher demand of getting rid of the waste, which is CO2. Higher amounts of thyroid hormone will also lead to an increase in GI motility. This means that there's going to be a higher secretion of digestive juices and motility of the food bolus through the GI tract. Hyperthyroidism is oftentimes associated with diarrhea, whereas when there's a lack of thyroid hormone, there can be constipation. There are also excitatory effects on the CNS. The increase of thyroid hormone increases the rapidity of cerebration, which can lead to psychoneurotic tendencies. This could mean anxiety complexes. You will also have irregular muscle contractions, and therefore one of the hallmark characteristics of hyperthyroidism is a fine muscle tremor. This can be assessed by a patient holding onto a piece of paper and seeing the shaking as a result of the paper undulating on their hand. This can also result in constant tiredness and fatigue, resulting in difficulty in sleeping. There are also several effects on other endocrine glands. Increasing of thyroid hormone can boost the increasing secretion rates of several other glands, noticeably the increasing of insulin via the pancreas. It also increases the rate by which adrenoglucocorticoids are inactivated by the liver. There's also going to be some sexual function effects. In normal sexual function, you need to have a balance of thyroid hormone. However, in men, when there's a lack of thyroid hormone, it can likely lead to a loss of libido, while a great amount of the hormone can cause potential impotence. In female, however, there's going to be a wider range of outcomes. When there's a lack of thyroid hormone, this could lead to menorrhagia or even polymenorrhea, which is excessive menstrual bleeding. Occasionally, in some females, there might be even amenorrhea, where there's an absence of menstrual bleeding completely.